evening, everybody. I hope you can all hear me well. Yes? Excellent. So as Alan said, I'm going to talk about scaling, but actually I'm not going to talk about that type of scaling. I'm going to talk about dynamical scaling and how we actually use that to be able to understand population dynamics. As Alan said, and my background is in zoology, I'm actually trained as an ecologist, so I'm going to talk a lot about ecology, but we also, as Alan said, in my group, we have lots of diverse interests across the breadth of the life sciences where we've been interested in thinking about applying mathematics. And I'm going to sort of delve into those sorts of things, and I'm also going to do a little experiment halfway through or thereabouts where some of you will get to eat chocolate, so you have to be fast. When I need 6 volunteers, the people who put their hands up fastest will get fed chocolate. So we're going to do that, and I'm going to sort of at the very end introduce another little game that we use to sort of explore some ideas in mathematics and biology. So this is what Alan was just saying. It's very, very, very common in biology. We think about scaling in a static way. So here, just like there's some sort of crab with a sort of a pincer, and we can relate the size of the body, the size of the crab to the size of its pincer. And often those are plotted on a log scale, and they show that this is very classic allometric scaling. And in this case that I just nicked from the web, we can just see there's actually the bigger the crab, the much bigger its pincer is in relation to its body size. So this idea of scaling, this allometric scaling in biology is very common, but it's a static thing. We tend just to measure species. We can go out and we can collect crabs, and we can measure the length of their carapaces, and we can measure the length of their pincers, and we can look to see how those relationships play out. But I'm a population dynamicist, and I'm interested in thinking about how things change through time and space, but tonight mostly about time. And we want to think about how we do that. How do we scale across these different types of dynamics? How do we take into account the fact that things may operate on different temporal or spatial, time or space, spatial scales? So I'm going to go through about 3 different types of ways that we approach this. And I'm going to sort of explain the maths, and I'm going to explain the biology behind some of these with some examples. I'm going to explain some of the maths. And at the end, I'm going to sort of talk about the implications, where this becomes really, really important, and why we think it's important in all the stuff that we do in extending our ideas of maths and biology to the real world. So I'm going to talk about slow-fast dynamics. I'm going to talk about this functional response around this point here, where we get to predator-prey interactions. That's where we get to eat chocolate. This point here, I'll talk about how we can use approximations, and I'll talk about some stuff that we've been doing in sort of thinking about how we can understand bipolar disorder and mood fluctuations, and I'm going to finish off with some implications and some very contemporary stuff that we do in terms of biological control. So slow-fast dynamics, what do I mean by that? Well, I'm thinking about these sorts of systems. So this is a cinnabar ragwort system. If any of you ride horses or know anything about horses, this plant is pretty nasty to horses. It causes lots of things that make them sick and poorly. It's a very common plant in the UK landscape. It's either an annual, short-lived perennial plant or long-lived perennial plant. It has these very colourful yellow flowers, but it's stuffed full of very nasty things that make horses and other things sick. This is the cinnabar moth. As a caterpillar, it feeds on this particular plant. It only feeds on this particular plant, and it's able to sequester, capture up those chemicals, and then these caterpillars, the caterpillars are distasteful and the moths are distasteful. This is very pretty. It's on the wing. It's out in about the middle of May through to the end of July, and it's a pretty common, as I said, a pretty common moth. We're interested in the dynamics. What sort of population dynamics? How does this moth and plant interact over time? And one of the things that we can do is that we can write down an equation to sort of capture that sort of dynamic. So this might be one way that we might do that. So I have resources are changing through time. So the change in resource through time on the left. And then I've got some biology that describes that sort of dynamic. So lambda is going to be some input rate of resources into the system. And then I have some consumption by the moths, which I've usefully called G. And they're consuming this resource at rate alpha. And the other thing that will happen is that if the resource doesn't get eaten, it will just decay away. And then there's some coupling, and I'll come back to that coupling later in the talk, but we're going to couple what gets eaten by the what gets eaten by the moths gets converted into new moths. So this is the birth rate of the moths, if you like, and the only thing that's going to happen to the moths is they're going to die. And we can do lots of analysis on this sort of model. We can really think about, you know, when the population dynamics would be stable, when they might show cycles. And that's quite involved. And we really want to think about, well, actually, if we're thinking about scaling, are those things operating on the same sort of temporal scales? And if not, can we get around that? Are there some approximations that we can take to think about that? So one of the things we can do is we can think, well, actually, maybe the resource dynamics are operating faster. Maybe this plant is just going through its life cycle a lot faster than the moth. And I can't really rub my tummy and pat my head at the same time, but you get the idea. This hand's going faster than this hand. So the resource dynamics are going faster than the moth dynamics. And the idea then is that the moth only experiences, on average, what the resources are doing. So we can then approximate those resource dynamics with a little bit of math. So we're going to assume that actually, on average, those resource dynamics are not changing. The dr by dt bit is set at 0. So if we do that, I can then sort of set that first equation equal to 0, and I can rearrange it just algebraically for r. And they're making a couple of assumptions in doing that. Firstly, as I said, the resource dynamics are going faster than the moth dynamics, but the other thing that we're going to make an assumption about is the loss rate, this decay rate of the resource. And we're just going to assume that the moths are really good at eating their resource and not so good, and they eat lots of it, so there's very little left around, so that actually the resource consumption rate is actually exceeding these loss rates. And if we do that, we can make this very simple approximation. And so if we make this rearrangement, so the average resource dynamics, R bar now, are given by lambda divided by the rate of consumption and the number of moths that are around. And that makes pretty good sense. Basically, if there's lots of resource flowing into the system, that means the resource is going to increase. And as the numbers of the moth increase, then the amount of resource is going to go down. So it captures, in some sense, what might happen to the resource, average amount of resource that's around in the system. But we're not really interested in that. I'm trying to think about, well, what would we do when we plug that into the second equation to do with the moth. So what I'm going to do is going to replace this term here by that approximation. So if I do that, I'm going to take this average rate of resource and replace the r in that second equation. And we get a much simpler expression now in terms of the moth dynamics. So I can basically say, I can cancel these two terms, and I'm left with lambda minus nu g. So basically, so in this slow fast dynamic, I have the amount of resource flowing into the system, and the other thing that's happening is that the moths are just dying. Well, it's a nice simple equation. It's a nice simple differential equation, and on the right hand side of this, and I'm pleasing my school teacher very much here, because she always used to tell me that I should show my workings a lot more. What we can do is that we can solve that equation. I can go from this equation to knowing what does this function actually look like for all time. So how many how many moths would be around at each point in time. So I can work through some calculus. Here's this equation written small, and here is my solution at the end. And it's a bit of calculus, and you need to know how to take the integral of 1 over g and something else, and it all falls out basically that I have this sort of function at the end. Okay, so that doesn't well, it may mean something to somebody, but we can do some things with this. Now I could draw it on the board, or we can just sort of do some thought experiments about what happens for different values of t. And the best values of t to pick are 0 and infinity. So if we set t equal to 0, and we work through this, and everything else is just we work through to see what happens, how many generators do we expect to be around at time 0, it turns out to be 0. So we're down at the origin of a graph. On an xy graph, we're down in that. So when there's no time, there's no moths around. If we set t to infinity, well, basically this term here then is essentially 0, and we're left with 1 minus 0 lambda over mu. Lambda over a nu. And what that basically tells me is that there's a fixed state. So if I know the value of the resource input, which is lambda, and I know this decay rate, then I should be able to say how many generators are around. But the very cool thing about this approximation is that we're setting t to infinity, and infinity goes on forever, then I go from 0 to some fixed constant value, a ratio between lambda and nu. And that's really cool because that's telling me that the moth dynamics should be pretty stable, right? So for all time, beyond 0, this graph is going to go up and it's going to stay flat for all time. And that tells me that actually with this approximation, my slow fast dynamics, I should expect to see resource dynamics being, the moth dynamics being relatively stable. Why is that? Well, there's an ecological process, so the resources are flowing into the system, and it's called the donor control. That's a bit of a jargon term, but it basically means that the resources are flowing into the system, and they're modulating the dynamics between the moth and the resource, and they're giving rise to these very stable dynamics. So the next thing we have to ask is, well, how true is that? Well, this is where data gets in the way. And we have the moths, so here's some pretty pictures of the moths, and this is the time series that was collected from the 1970s to the early 2000s of the cinnabar moth interaction. So I've got the log numbers on the y-axis and time over years. So every year, this person went out, they were based in the Netherlands, just south of Leiden, in some very nice habitat, and counting the number of ragwort plants and the number of moths on those plants over that time. So, and we see that the black line is the moths and the red dotted line is the plants. Okay, it doesn't really show that sort of expected dynamic. So we've got to think, well, why might that not be the case? And we'll get onto that very shortly. But it's actually sort of like brings in thinking about other bits of biology and and maths, and we want to think about, well, we don't see that nice stable interaction, that nice stable dynamics of the moths, and why is that? Well, we can appeal. We've got an idea about the sorts of dynamics that we have, and these time series that we have allow us to be able to do some statistical analysis. So I can ask basically what determines moth numbers in this place in the Netherlands, and how is it dependent? Is it dependent on the moths? Is it dependent on the resources? And how do those two things interact? So the black line is as I said, is the moth numbers. And I'm asking basically, how do those numbers change over time from one time step to the next? What are the drivers? How are the numbers in the previous time step important, or two time steps back, really important in driving these sorts of population dynamics? And we see two things. One, we see that the moths affect themselves, so they compete, if you like, for access to this plant in some way. But there's also a dependency of the plant. There's actually a link, that idea that they are specialists and they do need this plant, and that plant then drives their dynamics. But it's more interesting than that. It isn't actually the number of plants that are around in the last year, it's the number of plants that are around 2 years back that drives these sorts of dynamics of the moth. And we can do the same thing for the plants. We can do the time series and statistics on the plants, and we can say, how do the plant numbers change? And it turns out, well, yeah, plants affect themselves, both now, the number of plants now are affected by the numbers that were around last year and the numbers around in 2 years back, but also are affected by the number of moths that are around as well. So there's a really dynamic interaction there between the plants and the moths and the plant. And this approximation, while it's really good in theory, just sort of doesn't really hold when we compare to data because of this inherent link. And if I just flick back really quickly, it's this idea here, this idea of this response and this link between what gets eaten and what translates into new moths in the next time step. So that brings me really naturally onto this idea. We call that link in biology, in ecology, a functional response. If you're a biochemist, you'll know it as a Michaelis-Menten function, or it takes on many different words or phrases to explain what this is. But it's a link between two different states of the system. And it's a really great way, and we'll get into it shortly, about how you can approximate individual behavior and individual performance into dynamics. And the way I want to sort of explain that is with a completely different bit of biology, and that is to do with the blood stem cell system, the hemopoietic stem cell system. So we're really thinking about how the individual responses needed to make a blood system are dependent on what's around. It's called the demand control system, and your blood system is just fantastically fascinating. In your bone marrow, you have stem cells that are constantly making new blood cells, and they can make two broad types of stem cells. They can make two types of blood cells. The lymphoid cells, going down this sort of route, are all of the sorts of things that make your immune system, make it work. And the other ones are the sort of myeloid cells, which go on to make sort of a whole set of red blood cells. And we just think about it, we're just going to stylize that sort of interaction into thinking about myeloid cells, those that make red blood cells, and lymphoid cells that make immune system. But there's an immense amount of complexity in terms of the different types of cells that can be made. And obviously when it goes wrong, and it can make people very sick. And we're trying to understand how we can use our maths to actually understand the dynamics of this really incredibly diverse system, and how we can use these scalings, these functional response scalings, to understand that. So what we can do is that we can stylize it. We go from that sort of really complicated bit of biology to some sort of thing that I could write, just write out in terms of just a schematic diagram. So basically, we're thinking about transitions. So you can go, how do these stem cells give rise to new stem cells? How do they give rise to progenitor cells? And how do those progenitor cells give rise to those myeloid or those red blood cells or white blood cells? And what tends to happen, what the sort of ideas behind this that sort of reach back to the 1960s, we really just thought about two types of transitions that could happen. So this particular little blob represents a stem cell. And the things that can happen in the next time step, in the next event, are that I get two new stem cells. Or I can go from a stem cell to a progenitor cell that's going to give rise to a red blood cell or a white blood cell. But actually, the system's a lot more complicated than that. Look, if it's a demand control system, it's basically your body saying, I need more red blood cells, or I need more immune cells to fight off this disease. There has to be other things going on. And we can sort of extend that out. So a stem cell could say, there's a sort of like an emergency response that says, actually, this stem cell now needs to not only, it needs to sort of like die and make two new progenitor cells that then would make red or white blood cells, depending on what happened. The stem cell could die. The progenitor cell could differentiate even further and make two different types of progenitor cells, two more progenitor cells, because that's what happens in these stem cell systems. The progenitor cell could pass away, or it could differentiate into something that's really specialized, making a platelet or a T cell or whatever it might be. So we can really think about this sort of system in many different steps, and really many different sort of event-driven steps. And by that I mean that in any small interval of time, only one of these is allowed to occur. And the way that I'll show you what I mean by that shortly. But what we can do is that we're getting towards some more maths, we can sort of start to put some detail onto that. So here's the stem cell, here's a progenitor cell, and here's a differentiated cell, so a red or white blood cell. And these are sort of intermediate cells. So I have some background that affects whether the stem cells are going to give birth or they're going to die, or what controls their activity. But the important thing to note here is are these arrows. This one, so where we have these two particular functions, this negative signal. That's the demand control that we're interested in. We're really thinking about how do we link this, what's needed from the body in the peripheral blood to tell the stem cells what to do. Because they don't really know, they're just going to keep making things, and they need to be told what to do. And they need some way of linking that. And that's where that functional response actually comes in. So we've got progression of births, if you like, of progenitor cells, births of differentiated cells. But then when they've differentiated, we need to be able to understand, we need to be able to capture this idea of this negative feedback, this negative signal that says, make more or make less, do something, basically. We can capture that again with a set of ordinary differential equations, all of that biology, and they're more complicated than the ones I showed earlier. So again, we've got ds by dt, that's the change in stem cells through time. I've got dp by dt, that's the progenitors through time, and dd by dt is the change in differentiated cells. And then we've got bits of biology that capture the ideas of what we expect that happens in a stem cell system. So this first term, this term here, or this term here, depending on where, I hope you can see my point, is just telling us that the stem cell has a niche. It has a limited amount of size that it can occupy. Your bone marrow is limited in space, the space it occupies, and that means that the number of stem cells that can live in that space is going to be limited. And that's set by K. We call that the carrying capacity in ecology, but it's just a fixed amount of space that stem cells can live in. And then there's things that can happen. They can give birth to new stem cells, or they can change. They make progenitor cells, which is this term here. This is that second picture I had, where a stem cell replaces a stem cell and also makes a progenitor cell. Or this one here, where there's a 2, says there's a more emergency signal that says make me two, use a stem cell, need to now make me 2 progenitor cells. Okay? And then there's loss terms, there's decay terms, there's death terms of stem cells and death terms of progenitor cells, and transfer of progenitor cells into differentiated cells. But it's these 2 functions that are really critical at this functional response, at linking these dynamics, linking the individual performance to the population dynamics of the system. So if I use some numerical optimization methods, I can iterate that model. So here's the number of differentiated cells through time. The red line is the deterministic dynamic. So if I just run that model as a set of a coupled ordinary differential equations, I end up with this red line. As I said, it's a stochastic system. It's a random system. So if I make that event-driven, if I make each of those transitions an event that can only operate in a small time step, then I get a whole raft. I can sort of look at the sort of repertoire of dynamics that I see out of this. What do I expect from this system, given that it's random, knowing whether a stem cell is going to make another stem cell or a progenitor cell. And what this captures is actually the variability that we could see. It shows us that it's bounded in some sense, that actually the system doesn't go off to infinity and it doesn't go to 0. There's actually some bounds. So we can run this many times, and we can do some fancy maths, and we can quantify the variability that we see in the number of differentiated cells produced by any single one stem cell. And this is just a spatial plot, if you like, of just a heat plot for saying, well, here's the number of stem cells, sort of like around about 10, and here's the number of differentiated cells on average around about 40. But we see some spread around that. That functional response is, and that demand control is not perfect. Actually, and I'll come on to this shortly, is we need to understand in a little bit more detail this idea of homeostasis. And this idea of feedback, so how this demand feedback plays out, can occur in sort of 3 broad ways. So we're really thinking about the number of lymphoid, so a number of cells that are in peripheral blood. So we'll say the number of sort of immune cells that are around, and we think, well, what proportion of myeloid cells should be produced given this number of lymphoid cells? Well, the first one could be completely independent. These 2 things could just operate independently of each other. So we just have the number of myeloid cells. It doesn't really care how many immune cells are there. I'm just going to make more red blood cells because that's what I'm set up to do. Or there could be some relationship in proportional to need. So as there's more lymphoid cells, there's a need for more myeloid cells. Or it could be non-linear. It could actually start to increase and then plateau out. We need to be able to understand that. We need to be able to understand how this is sort of peripheral blood, how this peripheral blood feeds back its information to the stem cells. And this is the crux of it. We can write down a functional response. We can write down a mathematical expression that says how do we link the number or the proportion of cells that need to be made to what's already there. And we start by doing that in homeostasis, so where the system is not being perturbed. So we think of this, rho H, as the number of myeloid cells or the number of lymphoid cells that are being produced in homeostasis to maintain the body in a fixed state. And it's given by, so how many should I produce is in proportion to how many are there. This is the ratio of myeloid cells that are there to lymphoid cells that are there. And then there's some parameters, alpha and gamma, that we want to be able to understand what they look like and what value should they have that give us a homeostatic state. Well, if we want to know what our values are, we can sort of rearrange. If this is homeostasis, that's a fixed state. We know what that is because it's homeostasis. It's not changing over time. So I can then sort of rearrange this expression and say, well, what values of alpha give me this? What values of alpha should I have that give me this homeostatic state, and what values of gamma also give me this homeostatic state? And it turns out there's a whole family of them. They all look like this. There's a whole set of values. So here's the values of alpha and the values of gamma, and every single point on that line represents homeostasis. So there's a whole different value. There isn't one set of alpha and there isn't one gamma value that's going to tell me that this is homeostasis. It's just this whole family. Every value along this line is a point where we identify that will give us a homeostasis. Well, that's pretty cool, because actually what that means is I can pick a value of alpha and gamma and actually ask what would the functional response actually look like. It doesn't actually look, you know, how many, so what proportion of myeloid or lymphoid cells should I produce given how many are there? Basically, from each of those, choosing an alpha and an associated gamma gives me a whole set of family of outcomes to this. So the functional response, is it demand control? Is it in proportion to what's already there? Or is it independent of what's already there? Really depends on the parameter values that we have that are along that line. And we can get a whole set of different dynamical outcomes from this. And I'm about to explain what I mean by that and when we talk about predator-prey interactions. But we have a whole family, a whole set of different shape functions that relate individual performance, the demand required, the sort of like what's in peripheral blood, to what those stem cells should actually do, which is pretty cool. And I'm going to get on to talk about predator-prey interactions, and we're going to talk about the same sort of thing. So the same sort of thing plays out. We're really thinking about how do individual predators, moths eating plants or whatever it might be, polar bears eating seals or dinosaurs eating small rats or whatever it might be, how does that behavior of eating those things translate into a population dynamic outcome that affects these things through time? So again, another set of differential equations. You think, oh no, not another set of differential equations, but similar to the ones I introduced before. So I have different letters, but the same sort of biology. We're really good at this in biology. It does changing things with letters and not really changing much. But we've got predators and prey. So N is my numbers of prey, and P is my number of predators. And dN by dt is the change in prey, and dP by dt is the change in predators. And I've got some biology on the right-hand side. So I've got a birth rate, R. I've got a consumption rate by predators on prey. Now I've got a conversion rate, so I'm converting dead prey into new predators. And then I've got a death rate of predators. So let's just scale all that back and think, well, what sort of dynamics should I expect to have if the predators aren't there? So let's set P to 0 and see what happens here, basically. So we've got dN by dt Rn, basically. What sort of dynamics do we expect to see? So again, we can do some maths. This time, I'm not showing my workings, but I am showing a picture that shows the sorts of dynamics that we get. We expect if R is positive, so there's positive birth, that R is greater than 0, then the population of rabbits, in this case, will grow exponentially, unbounded, and they'll keep doing that, right? So that's all that's all we expect to see in this predator-prey in this interaction when the predators are not around. The population of rabbits, in my case, will just continue to expand through time, forever. Well, that's not going to really happen, because what's going to happen is that the predators are going to come along and start consuming rabbits, basically. And we're going to think, well, actually, we really want to understand how does it, as I said, we was really thinking about how does the individual predator eating a rabbit, which is a behavioral response, the predator has to go and find that rabbit and catch it and eat it, how does that translate into population dynamics? Okay. So we are going to work out what this functional response looks like and do some analysis. So here's the opportunity. I have 6 bags of chocolate. What you don't know is how many chocolates are in each bag. And I'd like to do this sort of experiment. It's a little exercise. If you don't, so I need 6 volunteers to put your hands up. I will pass you a tray, and I'll pass you a tray and a bag of chocolate, and we'll work through this. I can guarantee the trays came from the cafeteria, and they're still warm, so they're clean. I also bought clean pieces of paper as well, so feel free. I'm just going to put those there a moment. I'll hand out trays first. Take a tray. Who else? Can I give you a tray? Excellent. Can I give you a tray? Can I give you a tray? Excellent. All right. Anybody on this side want to try? Excellent. Oh, yeah, they're still wet. Oh, that's disgusting. Oh, I'm sorry. You're going to get a wet tray. Sorry. I've got one more. I'll go over this side of the room. Bear with me a moment. Somebody over here. Is it interesting enough? Excellent. Take a tray. It's a bit wet. Sorry. I'll be back with some chocolate in a moment. Right. All right. Right. All right. Now I have to remember where I gave my trays. Like a piece of paper. Thank you. And I'm going to give you a random bag of sweets. You've probably got about 25 in there. You need to open them and tip them onto the piece of paper. So I'm going to give you a piece of paper and a bag of chocolates. And what I need you to do when you have a piece of paper is to open the bag, sort of guess how many chocolates you have. I'll try and tell you. And just spread them out on the piece of paper. There's probably about 5 or 10 in there. I've got 2 up here. Somebody's going to get really lucky because one of these bags has got about 100 sweets in them. Woo! It's going to be a good day. Oh, I'm sorry, you've ducked out. You've got 5. Oh, I'm sorry. Don't worry. You could get worse. You've got about 100 in there. Where else is my tray? Oh, one here. Excellent. It's probably about 10 or 25 in there. So just okay, sorry. I have one more to do. Over here. You get the bag of M&Ms that's left in here, but you can't use them all, I'm afraid. I only need you to take 2 out of the bag, but you can share them with the people that are around you if you like. So there's the chocolates, and here's the bag of M&Ms. So just take 2 out and put them on the piece of paper. So if you've opened up your... So what we're going to do is we're going to do this idea of a functional response. Okay, and the idea is that you are going to be predators, and your prey are the M&Ms that you've cast out on your piece of paper. So tip them out onto the piece of paper. If you don't know the person next to you, it's a great opportunity to introduce yourself and just to help get them to sort of spread them at random so you can't see where they are. Because what we do, we're going to close our eyes, and you're going to take your predator claw, which is actually your index finger. This is your index finger. I have to say this every time. This is your index finger, and this is what you're going to use to search for your prey. Not your hand, your index finger, okay? And what you're going to do is you're going to close your eyes and you're going to tap on the board, on the piece of paper. When you find an M&M, please don't eat it just yet. Put it on the side because we need to know how many you've got. So have you all got a good idea of how many you've got on the paper? Yeah? How many you've got to start with? Yeah? How many have you got? 10? Somebody's got 100? Yeah? Somebody's got 50? Who's got 50? Stick your hand up. Who's got 50? Somebody must have quite a lot out there. Yeah? Yeah? So you've got some idea about how many you've got out there, right? Okay, so we're going to do this. So what the idea is that you're going to close your eyes, you're going to tap for a minute, you're going to see how many you find. Okay? And then what we're going to do is we're going to do some analysis, and we're going to see how your behavior of tapping relates to population dynamics. Okay, is everybody happy with that? Yeah? Cool? You all happy at the back? Yeah? Yeah? Everybody happy? I need to hear if everybody's happy. Wave if you're happy. Yeah? Cool? Excellent. Alan, have you got a watch? Alan, have you got a watch? Can you time me a minute? Yeah? Everyone ready? Excellent. Okay, is everybody ready? So you're going to close your eyes. Index fingers. People who hold them up. Let me see an index finger. Index fingers. Index fingers. Excellent. Okay. Close your eyes. Ready? We're going to tap for a minute. Go. How are we doing? Ten seconds. Excellent.
5, 4, 3, 2, 1.
Stop. Okay, please stop. Hopefully the person that found 2 managed to find them all. Yeah? Excellent. That's a good start. Okay. So we're going to do the analysis. Now you've found, so I just need you to count up how many you've found. Suppose somebody had 5. How many did you get? 5. Well done. You're a good predator. Excellent. Top of the chain. Excellent. Somebody had 10 on the board. How many did you find? 3. You're not so good, but that's okay. At least you've got them to eat for the rest of the lecture, even if I'm boring, so that's quite good. Somebody had 25 on the board. How many did you get? 22. Excellent. Did you use your index finger or your whole hand? Excellent. Good one. Always good to know. 22. All right. Somebody had 50. 38. 38. Wow, my God. This is going to be interesting. All right. So, and somebody, the person at the back? 42. Excellent. All righty, let's see how this works. So what we're going to do, I am going to quickly make sure I've done this right. Ah, there we go. Always an issue when it doesn't like the numbers, but hey, it's got a bit cranky. It's not going to work. Okay, it's not going to work. So what okay, I'm just going to fudge the numbers, but you'll see just a little bit, just to make it look. Let's see if we can make this. Just to give you an idea of what happens. So the two things that go, there are 2 types of responses that go on. So I have a picture somewhere here. So you guys were really great. Without a shadow of doubt, my analysis doesn't take into account the fact that you could be linear. The fact is that what we have on this graph, and sorry, the font is not great. On the x-axis, I have the number of M&Ms that are on the board. And on the y-axis, I have the number that were caught in a minute. And what you guys have done is managed to find, whether you have 100 on the board, you've found half of them. When you have 50 on the board, you've got quite a lot. And the idea is that that response is linear. It will go, I've fudged the points because we're trying to fit a curve through this. But you would have a nice linear relationship there of the number caught and the number on the board and the number that you caught. What tends to happen when we do this and we replicate, and obviously I can only do this with 6 of you, if you all were able to do this and we were in a little bit more of a controlled environment, when we were doing this experiment, what would happen is that over time, even when you had, how many did you find when you had 100 on the board? 42? 42, right? So you don't find them all, right? You find about half of them. And the really important thing there is actually what we're doing is we're linking the behavior, this idea of foraging for the prey, to the population dynamics. And what we expect to see is that as you increase on the x-axis, you can't actually get many more than about 40 out of 30 or something out of this. There's a finite amount of time that you have to actually be able to take these M&Ms off the board and put them on the side, or as a predator, kill your zebra and convert that zebra into a new lion. That takes time. You have to eat the thing, you have to digest it, you have to give birth to new lions. That's a time-dependent response, and it has this particular shape. Okay? So as the number increases, so I have numbers up on this axis at the moment against the numbers on the board. But if I think, instead of having numbers, if I have the proportion, what happens with this graph is that actually down at the low end, most things get caught. That might be counterintuitive, but actually, if there's a small number of prey, most of them are going to get captured. But when there's a large number of prey, they don't all get caught, and as the number increases, they continues to go down. So we have this negative relationship. So the proportion eaten against the number on the board goes down, and that basically means out here, when we have lots of prey, then we expect to see many of those prey are escaping predation and are able then to do whatever they do, give birth. So we expect to see that actually when there's a large number of prey, when the small number of prey are being caught, lots of them will be able to increase, and in the absence of predation, as we showed earlier, they will increase exponentially. So how does that translate to my moth dynamics? So I have this sort of idea. I have this sort of relationship that actually, when we have this dynamic, when we have this functional response between behavior and dynamics, they give rise to this idea that when there's lots of them around, proportionally very few of them are going to get eaten, lots of them will escape predation, means that they can grow exponentially, and then what happens is that the predator catches up. It's a numerical game. So if there's more prey around, there's going to be eventually more predators around, and then that's going to turn over, and we get these predator-prey type cycles. And that's what we see in this system, basically. If we see again, this is the moth numbers through time. The black line is the moths, the red dotted line is the plants. And we see this cyclic dynamic playing out. And that's because of the relationship, the link between, the scaling between individual performance, how a predator, or a moth in this case, performs, and how that translates into the population dynamics. And that's pretty cool. It's a pretty cool way of which linking, different aspects. So behavior operates on a different temporal scale to population dynamics. And when we can take that, we can translate it into these sorts of population dynamics. So I want to finish. I want to sort of talk about the third way that we can sort of think about scalings, and it's a bit mathematical, but it's about approximations. How we can approximate dynamics that operate on a different time scale. It's like the fast-slow dynamics, but we can do it in a lot better and a little bit more rigorous way. And I want to sort of motivate this by a really useful and sort of really cool way in which we approach this sort of analysis. We are interested, and a group of colleagues in Oxford and elsewhere, we're interested in being able to understand the mood dynamics associated with patients with bipolar disorder. So bipolar, very common mental illness that occurs in sort of late teens, early 20s, and it's sort of lifelong chronic condition where you get episodes of mania and depression. But in between, those episodes of mania and depression are really, really rare compared to the mood fluctuations that go on a day-to-day or a week-to-week basis. And what we can use, there's a set of standardized psychological scoring standardized psychological scoring metrics that can be used to quantify mood. And they're called QIDS. So it's a quick inventory of depressed symptoms. And we can use that. It's a self-reporting system. People use their cell phones or the computer to answer a set of 16 questions that give an answer to, and we can create an aggregated score out of that. So this is what this is. These are the QIDS scores. Those are the mood scores that we get out of patients over a period of time. And we can do that. We can do that on different temporal scales. We can do that on a daily basis. We can do it on a weekly basis. We can do it before and after intervention. And this is just a sort of cohort experiment that we had for 15 patients or so, where we monitored their mood, which is the solid black line, over a 28-day period before and after a treatment intervention. My colleagues I work with this are clinical psychologists, and they use behavioral interventions, so cognitive behavioral therapy, to treat people and help people with bipolar disorder. And what we can do is we can ask the question, does their mood fluctuations change after treatment? So we can quantify it before, they go into treatment for 4 weeks, and they come out the other side, and we can characterize their mood dynamics. So this is what we see. We're able to sort of characterize these mood profiles and just collect them in a temporal time series sequence. So again, it's just population dynamics. It's just population ecology, the sort of stuff I grew up doing, sort of with a very clinical relevance here. So what we can do is that we can sort of pick that apart a little bit, and we can ask basically, how can we understand mood dynamics? How can we get to the maths, mood maths we call it? How can we unpick the dynamical things that drive mood fluctuations? And we've done a bunch of things. We've used a whole set of different mathematical methods, and I'm just going to sort of talk to you about this one and how we can then link this to some scaling dynamics, so how we scale between different things that may be going on. So again, another just sort of differential equation. We're going to sort of split mood up into sort of we'll take the words at the moment. I've got change in mood, so from one time step to the next, a very small time step. I'm not going to be explicit about what that is at the moment. And then we can split that up, and we can use a total derivative approach. We can split it up into different component parts, essentially. So I've got some sort of partial derivative of mood with respect to time, so that might just be baseline mood. What happens, what just happens to your mood in just baseline? And then we can sort of split it up further into something to do with fluctuations. Something's driving this mood fluctuations in our heads, and we've got to really think about how we understand that. And we can sort of split that. We can sort of think of this dx by dt and dz by dt as oscillators, something that's driving that. So they may be inhibitory and accumulating oscillators that drive dynamics. And then there's sort of these sorts of things, the partial change in mood with respect to x is just a slope. It's just a regression on how mood changes with respect to the size of the oscillator that we're, or the inhibitor or excitatory effect that we're trying to measure. So we can write out this differential equation in terms of the change in mood is just baseline mood. That's just going to be flat. It's just some constant that's going to change over. That's not going to change over time. And then these things are going to be fit. So there's going to be fluctuations to do with this oscillator and this oscillator with some magnitude associated with the parameters in front of that. And what we want to be able to do, and we can do that statistically, we've done that statistically, and that was what was on the previous slide, is that we can try and fit these sorts of models to the time series. But I want to get into a little bit more about this, and there's a lot of words and there's a lot of detail on this slide. And what I want to just explain is that we can write this differential equation, or this differential equation, as an oscillator. So we're thinking about things that inhibit and things that accumulate, sort of like these dynamical fluctuations. And the really cool way, one of the really cool ways to be able to capture that, and phenomenologically, very broadly, without too much mechanistic detail, is with what we call a relaxation oscillator. And a relaxation oscillator is a really cool thing. It's just it's going to oscillate, it's going to go up and down through time. And it's relaxation because actually I'm going to spend a long time in one state and I'm going to relax very quickly to another state. So it's just like a predator-prey cycle. That might be one way to think of it, is that I'm going to stay in a high state and I'm going to stay there, and then I'm going to relax very quickly to a low state, and then I'm going to relax out of that state to a high state again. And we can capture that. We can capture that phenomenologically, very broadly, with this set of ordinary differential equations. So we can couple two things together. We can put some complicated nonlinear function in there, and then we can capture this idea of a relaxation oscillator. Well, for bipolar disorder, that's pretty cool, because then we can think about that as actually you're in a state, maybe you're in a high mood state, and you can relax to a baseline state. You may be in a low mood state, and you can relax to a baseline state. So we're capturing that idea. That might be one idea that we're trying to capture. We may also be trying to capture this idea that there's inhibition and excitatory processes going on within our heads to sort of drive the dynamics. We can do other things. So there's a little bit more complicated stuff on this slide. There's some stuff at the end. So this stuff here and this stuff here is capturing the idea that we can make these things stochastic. So we can kick the system with a little bit of randomness at each time step just to add some additional fluctuations in that may not be captured very broadly by this oscillator. And the other cool thing we can do is that we can couple these things together. We can say, actually, what's happening to one isn't independent of what is happening in the other. So we can really make this sort of scaling. And we can, as I said, we can fit that to the time series. So we can introduce noise, and we can look at all of these couplings, and we can challenge these models against the data and look at what the explanations might be that best describe individual patients. So you, as an individual patient with bipolar, do you need two oscillators? Do they have to be coupled? Do they have to be noisy? And it really is, and it really helps us understand the sort of temporal dynamics of bipolar. So here's just what we do. We can sort of face 3 patients just picked at random, and here's their oscillators. So they're all oscillating. They don't have to oscillate. The parameter values, the parameters in that oscillator could actually give rise to stable dynamics. It's just what values they choose to take on when we fit them to the time series. But each of them, as you can see, each of these 3 patients has an idiosyncratic sort of like oscillator system. Sometimes they have to be coupled, sometimes they don't, sometimes they're not, sometimes they have to be noisy, sometimes they don't. And we can fit those to the time series, and you can see how good a fit they are, which is the purple dots in this case. It's a really good technical challenge, because actually bipolar patients are pretty bad at reporting every time, so there's lots of missing values in this, and we have to be able to deal with those statistically, and that's all pretty cool too. But we can do that, and it's pretty nice. But that's not what I want to get to. I want to take you to where we are with all of this, because I think it's really cool. Because what we're trying to do is really understand. So I've explained this. This is my total derivative. I've got the baseline mood. I've got some parameter associated with these fluctuations. What we're really trying to do is to step down into heads and think about how do neuron firing patterns drive mood dynamics. That's a huge ask, right? We're actually neuron firing dynamics are operating on such a fast time scale compared to how fast your mood changes. I don't know like my mood, but my mood's, well, I don't know, up and down at the moment because I'm standing here talking, but, you know, it changes, you know, not as fast as, and my neurons are going like, oh my God, I've got to keep talking. They're really firing off in my head to help hopefully make me lucid. But what we really want to do is to link these sorts of neuron firing patterns to this population dynamic, this dynamic of mood change over time. And these are operating on different timescales. And the way that we're thinking about approaching that is actually approximating these functions. So these two differential equations are describing the inhibitory and excitatory processes that go on in neuron firing patterns. And we can aggregate those together, and we can really think about how we can describe. We can just take those two differential equations, these two here, and really get into the neurobiology and the theoretical neurobiology and the math neurobiology of that. But we want to take it up a scale. But these are operating on different timescales, and the way that we can do that is to approximate these things with a Taylor series. And a Taylor series is just a way of writing out a function in an infinite sum, and we can write it out in terms of at a particular point in time. And we can take that function and we can just say, well, what is its linear component? And then what is its quadratic component? And what is its cubic component? And what is its quartic component? And we can just keep adding on forever. And we can get an understanding, we can approximate these neuron dynamics at a particular point in the mood dynamics with this Taylor expansion. And that holds a great possibility. It's where we are, we're at this step, and this is where I've taken you to where we are in this sort of problem. I don't have any answers to that yet, but we think it's a really cool approach to be able to approximate the neuron dynamics in terms of using these sorts of Taylor expansions. So let me finish up with another sort of implication about scaling. A different one, completely different one. Just another little game we'll play very shortly. But it's to do with sort of like thinking about biocontrol. People in my group, who I guess are sat around here, are interested in thinking about how we control pests, insect pests, vectors that spread disease, how we can think about the biological control of doing that, and how maths helps us understand that. There's a very old method in insect control called the sterile insect technique. And the idea is that you would mass rear these insects, zap them with radiation, make them sterile, release them out into the environment, and when they make no viable offspring. So the eggs don't hatch, or the insects die, or whatever it might be. So the idea being that you're aiming to sort of like send the population into decline because the matings are not successful. Well, there are many new twists on that, and one of the latest ones is being able to use genetics to do that. So you can genetically modify your favorite agricultural pest or your mosquito that's spreading disease. You can change its genes so that when you mass rear them again, throw them out, when they mate with wild types, they don't make any viable offspring. Those larvae may die, or the caterpillars may die, or the eggs may die. So the idea being that you're trying to suppress the population. We can do it both conventionally with radiation or with genetics. So the idea is essentially to suppress the population. There's a whole set of cool maths behind all of that, and it's to do with what we think about as optimal control. We can do optimization, we can do static optimization, we can say actually we want to just know what this is for some fixed values, but actually this is a dynamic problem. And somebody said you should shock people, Mike, so here's the shocking bit of this. Still, so this is a bit of an old poster, but actually still something like 2,000 kids die every day from malaria. Since I've been speaking over the last hour, probably 50 kids have died from malaria. But it's a completely preventable disease, and using these sorts of methods, these biocontrol methods, we have an opportunity to be able to do that. And our maths really helps us think about that, because actually we've got a dynamic, so this equation at the top, dv by dt, is the change in the vector numbers, the change in the number of mosquitoes over time. And it's something to do, there's some births of mosquitoes and some deaths of mosquitoes. And we can fact this thing in brackets is the function that says, I'm going to do this sterile insect release. I'm either going to do it genetically or I'm going to do it conventionally, and I'm going to release a number of modified mosquitoes, I. Well, if I is 0, V over Vt is just going to be 1, so it's going to cancel out. But it's actually, as I increase the number of these sterile releases, this term is going to become more important, and this term is going to, and the whole term on the first term on the right-hand side is going to get smaller. We're aiming to reduce the number of births. But it's not all that's what's happening in this system when we're thinking about malaria. that we've got people being infected, and we've got infected vectors. So we've got a dynamic situation there. We've got dynamics operating in terms of the changes in the vector numbers being born and being controlled. What we're trying to do, we're trying to stop people getting sick. So actually, we can think about this as an optimal control problem. This is a dynamic control problem, in that we're thinking about actually what we want to do is we want to control this problem over time, a fixed period of time. We're going to discount it. That's an economics term. It's a way of scaling, thinking the future. And we're going to think about what we want to do is we want to minimize this function in terms of the number of people that are getting sick and the number of the number of these sterile insects that we have to put out there. So that's an optimization. This has to be bigger than 0, but we don't want it to be too bigger than 0 because that's going to cost us. It's going to be really costly to do that. Economically, it's going to be costly to do that. And this is where we're able to scale both biology and economics. And what I'd like to do, just to finish up, is just to on our group website. It's just going to work. We have a game, and we can play this game. And we'll just play this game because it's really instructive. It's an optimal control game, and it's sort of set out without any of the math detail. It's got lots of the biology in there. So one of the first things you need to do when you're thinking about this economics of biocontrol is to know the problem, know what the size of the problem is that you're trying to control. And the way that we do that is we have to estimate what the pest population or the vector population is. How big is it? Well, ecologists do that with a method called the mark, release, recapture method. So you mark a bunch of insects, you throw them out, you wait a bit of time, you go back and you collect a bunch and you work out the proportion that were marked versus unmarked, and it tells you you can use some wacky formula, and it tells you the number of mosquitoes. Oh, that was the wrong one. Damn. Let me just go back. Did you do the mark recapture? No, I did not. Okay, so we're just not going to do it because with kids, little kids, we get them to run around and they can find mosquitoes and we can do all this stuff. So we're going to pick a number of how many mosquitoes we want in the population. There's some stuff about the biology, but this is where it becomes important. We're trying to optimize this problem. We're trying to make it as cost effective as possible. We're trying to scale the economics and the biology to make this as cost effective as possible. And there are two things. There are two things you can control. Firstly, how many sterile mosquitoes, how many sterile insects am I going to put out there? And then I'm going to think, how long can I run my program for? So the top one is the ecology, and the bottom one is the economics. Okay? So on the top one, I can choose any number between 1 and 10. Choose me a number between 1 and 10. 2. That's a good number. Okay, so that means for every wild type mosquito I'm going to put out there, I'm going to release 2 sterile insects. Okay? How long do you want to run your program for? You can have 2, 5, or 10.
5.
Oh, 5 was the first number I heard. That's a good number. Okay, so we'll see what happens. So we're going to get 2 graphs. One of them, on the top one, we're going to do the ecology of the mosquito, the population dynamics of the mosquito. On the bottom one, we're going to see how many people you managed to save with your particular program. Okay? And we're going to look for a dollar figure. And we want that to be as small as possible. The bigger it is, the worse it's going to be, essentially. So I'm going to start, and the other bit of ecology is that I need to know when in the season I'm going to start my control. I'm just going to pick one of these. I'm going to see 2 lines, basically. And then the black line on the top is what I did in the absence of control, and the green line is what I do in the presence of control. And you can see that actually I can reduce my... So I'm doing a 2-to-1 release ratio, so 2 sterile mosquitoes to every wild type. And on the bottom, I have the number of sick people in red, that in the absence of control, and in the presence of control in blue. And it goes up and down because we're thinking about mosquitoes and they like it wet and dry, or they like it wet and they don't like it dry, so there's some seasonal dynamics there, essentially. One of the very cool things about this is actually, we haven't quite got to the numbers yet, but let's talk about the dynamics, is that on the top, I only suppress my population a little bit. This 2-to-1 release ratio doesn't actually squash the population down very far at all. Yeah, it's only been suppressed a little bit. But actually, I can reduce the disease dynamics by about half. Yeah? The average number between the red line and the blue line has gone down by half. So that's pretty cool, right? Actually, I do not... So one of the things is I don't actually need to eradicate my mosquitoes from the system to start to have an effect on disease control. If I choose a bigger number, it would be more costly to do that, and actually I could get the same sort of phenomena. And as we know, this is a well-known fact in epidemiology, where if you suppress those agricultural pests or those disease vectors below a threshold, then You know, you don't have to eradicate them, you just have to push them down below that threshold, then you can sort of have a big effect on the disease dynamics. We stop our control after 5 years, we run out of cash, so that stops after 5 years, and actually then, because of reinvasion, there's really good documented evidence of that happening throughout the world, that actually we see the disease rebounding to pre-control levels. So what happens? The most important thing is what this dollar figure is at the bottom. How many cases did we avert? How many cases did we avoid? And how much did it cost? How much was the control in that? What was the optimal value that we can get out of that? Turns out 252 is not bad, but we can be better. I had a 7-year-old girl do this one time. She spent 45 minutes playing on this game, and she came out with 229. I've never got down to 229. So if anybody plays this game and gets below 229, I want to know. Because it's really important thinking about how we are able to blend this idea of economics and ecology and scaling these two things. Because they're different problems. They're operating on different timescales. You've got to think about how long you run your program for, and that could be many years. But these mosquitoes and people getting sick is on a daily basis. And as I said, many people still die from these sorts of infectious diseases. Okay, I am just about done. I think, okay, I've told you about 3 things. 3 ways that we can scale slow fast dynamics. I didn't do the scaling laws that Alan might have expected to do, but I've done some dynamical stuff, and I hope you found it interesting. We can do slow fast dynamics, so we can set those things, one going faster than the other, and we can make really cool approximations and use some very, very cool maths to work that through. We worked on the functional response, predator-prey interactions in particular, and there's ways in which we can approximate these things with particular types of functions like the Taylor series. There's really nice ways in linking things that operate on different timescales. We do a raft of things in my group. If anybody's interested, we have a group website. Some of them are around the room. If they stick their hands up, they may show their faces, they may not. They may run out of the room thinking, my God, my supervisor's embarrassing me, but that's perfectly fine. And I'm more than happy to take some questions. Thank you.